Okay, so I work on theoretical population genetics, that is theoretical models of evolution, and I'll present some work that I have done with my PhD student Archana Devi, who is also in the audience. So before I do that, let me start from the starting. So what is a biological evolution? What does it mean? So the crux of the matter is just in these 3-4 lines. Let me explain what it means. It just says that, in a population, there is variation, as you can see in this room. And if these variants, they are good in this particular environment, they will be selected. And this selected variants will be passed on from one generation to the next. Okay, so and then the process would basically repeat. Okay, so that's what natural selection is doing. It is going to reject that which is bad and preserving and adding up all that is good. So that's the sort of basic funda. But Darwin believed that this phenomenon, which is creating all this diversity of life, it is not seeable. It is such a slow process that we can't observe it. And that, as a physicist or maybe as a biologist, is a very unsatisfactory thing. So this was a sort of paradigm that it happens, but you can't really see it. But this has changed a lot. This idea that it's a very slow process, this has changed over the last 40, 50 years. Nowadays, we find lots and lots of examples in which you can see evolution right in action. So there are lots of examples, and I'll just give you one of them. So here is this classic experiment that started in Richard Lenski's lab. So he started this experiment in 1988, and it's still going on. So the idea is very simple. It's just that he has this 10 flasks of E. coli, and all that he has done is to give them less glucose than what they're used to. And then this population has to make mutations. Something should happen at the genetic level so that it can adapt to this bad environment in which it is getting less food. So what is seen is then this fitness, measured in a certain way, it changes over time, it increases over time. And you can see it's a very fast change. Over just 10,000 generations, there's a 40% change. It slows down and so on. Okay? So therefore, evolution is very much observable. So that's one point I wanted to make. Secondly, things also have changed in another way. That is, one now does not think just evolution as one doesn't explain this in a qualitative manner, saying fitness increases, that's qualitative. In last 10, 15 years, there's a real effort to understand these things in a quantitative manner. So in fact, these two curves that you see, green curve and the red curve, they are from certain theory. So people are actually trying to understand these numbers now. Okay, fine. Okay, so what are these models? So I wanted to give you some idea about this. So I'll describe this one model, which is the sort of standard model which any PopGen book, if you open, you will see this. It's called the Wright-Fisher process. So the idea here is this, that I have a population, let's say of 5 individuals, then each of them carry with them a number called their fitness. You expect, intuitively, that a parent who has a high fitness would have larger number of offspring. Lower fitness will have lower offspring, and so on. But since the system has a finite carrying capacity, you will get lots of offspring, and then you sort of, remove half of them or, some number so that according to the carrying capacity of the system. So one way of implementing that is to say that the offspring chooses a parent with a probability proportional to the parent's fitness. So that's how these, these lines or links have been drawn. So time is going forward, and some individuals have 0 offspring, some have 4 offspring, although all of them happen to have the same fitness. So this is the stochastic evolution. So this population is, doing its thing, and sometime a mutant appears. And then it does the same kind of dynamics as this light-colored wild type ones, except that it has maybe a higher fitness or a lower fitness. So let's look at this mutant for a second, because that's the one which is going to drive this whole process of evolution. If everybody is same, there's no variation, there's no evolution, right? So therefore, now mutant has appeared, and we need to focus on that. Okay, so there are some stochastic trajectories that I'm showing you for the model, for the Wright-Fisher process, and I'm just looking at how the number of these dark circles changing over time. So these are just some trajectories. So you can see that it started as a single copy. The mutant came in a single individual. Then in some trajectories, after some time, it does some drifts, and then it gets lost in some of the trajectories. Okay? In other ones, it can reach a frequency 1. This reminds you of something that you have seen even yesterday. These are absorbing states. So let me repeat this point a bit. So here I'm saying that there's a single copy which came in. Then because mutations occur rarely, so all can become just dark circles. The reverse thing is rare. So basically for practical purposes, at least over some time scales, this is an absorbing state. And similarly, they all can become empty circles, and that's also an absorbing state. So unlike the talks that you heard yesterday, there are 2 absorbing states. And before I can ask questions about first passage time and so on, the first thing I need to ask is, what's the chance that I wind up into one of these absorbing states? Okay, so that's what one can do. And one can write down a standard backward Fokker-Planck equation. So this is the time that I start. So I start with x0 frequency at time 0, and I want to know what's the frequency at time t. It's x. The frequency is x at time t. And one writes a backward equation, which is slightly different from the usual ones, in the sense that these coefficients are coming outside the derivatives. So this is the deterministic term, which is logistic. Okay? And this term is not a diffusion constant. First of all, it's not a constant. Also, if you write in the forward equation, it comes inside the second derivative. So you should not think of it as a diffusion constant, which is space dependent or something. Okay? So this is very different from what we are usually used to in seeing in physics. Okay. Typically, one is interested in asking what happens at large times, and basically dp by dt is 0, and you can easily solve this simple ODE and obtain this classical, very celebrated formula due to a population geneticist called Mutu Kimura. Okay, and one can also work out times of fixation and so on. Okay, so that's a sort of introduction to what I wanted to say. So here, this whole thing I have assumed that the selection is constant in time. But of course, environments change. So one would like to know what does that do to these fixation probabilities. So that's what I want to address now. And I'm particularly looking at the case in which selection or these growth rates change periodically with time. So again, we write down the corresponding Fokker-Planck equation. At large times also, in the previous one, this derivative dropped out. But now if you write down, you get 2 time derivatives, one for the final time and one for the initial time. So there are 2 differences from the standard one. First, there is this logistic growth which depends on time, and also the fixation probability depends on the arrival time. This equation is inhomogeneous in time. Okay, so you still have to analyze this PDE. Okay, so now how to do that? So we can figure it out, but let me just see what's the qualitative picture. What do we expect? So there is, environment is changing at a rate omega. So if omega is very small, it's more or less like a static environment. So if I just show you the trajectories, the environment has changed from slightly below 0.6 to 0.6, say. Then the fixation happens. So fixation means the frequency became 1. It happens much faster, much earlier than the rate, the time over which environment changes. So it's more like static problem, and you can sort of cook up some static, adiabatic approximations. That's what one thinks that one could do. Okay, what happens in the opposite limit when the environment is changing very rapidly? So tick 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 is changing very fast, and then the frequency, sees what kind of environment. So imagine this. So therefore, now I'm a mutant. I see good environment. I try to increase my frequency, but it's changed. I mean, before I could do that, right? And I have to reduce the frequency. So if the environment is changing very rapidly, the mutant actually sees just the time average environment. Okay? And in this case, because I said it's a Fokker-Planck equation which is inhomogeneous in time, which depends on the arrival time, here there's no memory of the initial phase, at least in this omega going to infinity limit. But then maybe I'm just changing it too fast. So let me check sort of large but finite rate of change. And here you see at short times, the frequency changes quite drastically. It gets nearly 0. But once one cycle is over, it doesn't crash quite as much. In other words, at large but finite frequencies also, I expect an initial time dependence. Okay, so now I need to put this together. I can sort of know what to expect in a low frequency, slowly changing environments. I sort of expect I know sort of what to expect very fast changing ones, but we would like to write down some simple formulas by analyzing this PDE. So we did that using a time-dependent perturbation theory. I'm not aware of any answers, exact solutions to that kind of equations. So if anybody knows, I'll be happy to talk to you. But anyway, so we did a perturbation theory for very small omega, large omega, and we see that there is this initial phase dependence simply cos theta with other factors and so on. Okay, so here is the from the these are the theoretical analytical answers, and these are from numerics from the two sides. So the thing is that this fixation probability is non-monotonic. There is a resonant frequency below which and above which one can get some simple answers. Okay, and at this resonant frequency, the chances of success are larger than they were in the static environment. But it's not always a good news. Answer depends on the arrival time. So here I chose a certain arrival time in which fixation probability was maximized. For different case, it turns out there's a minimum in the fixation probability. Okay, so one can sort of understand, and it has evolutionary implications. But okay, let me just finish it. So here's the thing. So there's a couple of simple points. So I'm just saying that in a dynamic environment, chances of success, they can be substantially different from what you expect in the static environment. Okay, and one has sort of characterized that quantitatively. And these answers depend on how fast environment is changing, and also depends on the time at which the mutant arose. And this was a simple example of inhomogeneous backward Fokker-Planck equation. Okay, thank you.